All right, we are starting the recording. There is so much more we could talk about as far as DNA goes. There's this whole new field that's opened up of molecular biology, genetic engineering, therapies for diseases that have opened up new approaches to developing, well, things like vaccines and treatments. We talked a little bit about that. There's an entire sector of, the economy, the biotechnology industry that didn't even exist when I was born. Certainly, this is a major frontier. Unfortunately, I've got to finish the course. So hopefully you've got enough background that if you need to know more about fields like that, you can move into them. And we've got one more area of biology to cover in the next two lectures. So let's go ahead and get a move on. All right. And it's the field of ecology, but I'm going to get there by talking a little bit more about celling. The idea that there's two basic types of cell. Eukaryotes have compartments, one of which is a compartment for the DNA called the nucleus, and other compartments that do various things called the organelles. And prokaryotes generally are not as compartmentalized. Now we can go back and look at this in a little bit more detail. This picture shows you a single-celled eukaryote. It's called an amoeba. Some of these can get up to almost a millimeter long, so these are very large cells. It's in the process of engulfing a different eukaryote, a single-celled protist called Paramecium bursaria. That's the greenish thing to the right. And any cell, prokaryote or eukaryote, contains a liquid substance called cytoplasm. If you could see this amoeba moving, you would see the cytoplasm flowing within it. An amoeba is basically a giant mobile blob that can send out projections of itself and withdraw projections into itself and move through a process of cytoplasmic flow. Cytoplasm is a complex solution of water, of course, with lots of salts, lots of proteins, various other substances, and everything inside the cell is floating or is suspended inside the cytoplasm. So the cytoplasm is the liquid that contains all that grainy stuff inside this amoeba, which, by the way, there's what looks like an empty bubble in the middle of this amoeba. That's not the nucleus, but the nucleus is just down and to the left of that empty bubble. That empty bubble is called a vacuole. We'll talk about that in a bit. What surrounds the cytoplasm is called the plasma membrane, and this is actually made of lipids. You might remember way back we talked about carbohydrates, lipids, proteins, and nucleic acids. You've got a lab to do about those, one of the Labster simulations. Lipids is the class of macromolecules that includes fats and oils, also waxes, incidentally. And a major thing that lipids do is literally form membranes. The membrane that keeps the cytoplasm in and the outside outside. Specifically, this is a type of lipid called a phospholipid. By the Allfather, do not try to memorize all of the stuff that's here on the diagram. What I want you to see is that proteins and other molecules are actually embedded in the membrane. They may stick out of one side or the other or both. So at the far left of this diagram, this thing labeled the protein channel, that's a tube-shaped protein that can open and close to let some substances into the cell, but not others. There are other proteins that act as receptors. They will bridge the membrane, and part will stick outside, and then that part can respond to something, some stimulus from the outside, and change its shape on the inside and enable the cell to make some kind of response. There's lots of others. Don't worry about all this detail. The membrane isn't really solid. It's actually a very thin liquid film, more like a soap bubble than anything else. If you've ever looked carefully at a soap bubble, especially in sunlight, you may have seen that fluid, can flow, make little swirling patterns and things like that. But it's restricted to the surface of the soap bubble. You can think of membranes as being like that. A thin film of liquid, but one that is only free to flow in 2 dimensions. That is free to flow, along the dimensions of the surface. And since cell membranes are 2-dimensional liquid films, it's easy for them to fuse together or to pinch apart. You saw some of that pinching apart when we talked about cell division, cytokinesis, and all of that. Cell membranes can also join and come together. You'll see some cases of that happening in just a little bit. And you could think of this as being analogous to bubbles in soap suds. If you've ever looked at, soap suds in your sink, you might actually see 2 small bubbles fuse together and suddenly form a big one. If you haven't, try it sometime. It's not that hard to observe. Cell membranes can do similar tricks. They can fuse with each other. They can, pinch apart, like one large bubble dividing into 2, which you might see if you've ever blown bubbles with a bubble wand. Yeah, they're these permanent liquid films that can join and rejoin and split. Okay, great. Prokaryotes have a cell wall outside the cell membrane. We talked about that. There's a central region inside the cell called the nucleoid, where the DNA is located, but it's not bounded by a membrane sac. It's basically just a heap of DNA sitting there in the middle, but there's no membrane that makes a barrier between the DNA and the rest of the cell. You can actually see this. This is a slice through a bacterium imaged with a transmission electron microscope, and the nucleoid is just that white stringy area in the middle. Ribosomes, even at this scale, they're just visible as little flecks, little black dots in here, those little black grains that fill up the prokaryote cytoplasm. Those are ribosomes, and they're pretty much just floating there freely. And prokaryotes don't have chromosomes the way that eukaryotes do, so they don't divide by mitosis and meiosis. A chromosome in a eukaryote is basically one long strip of DNA. It's got one end and it's got another end. Prokaryotes, bacteria and archaea, have DNA that's organized into one large loop, plus a number of smaller loops. This is an electron micrograph of some of these small loops of DNA. You're actually seeing individual DNA molecules here, and those loops are called plasmids. And bacteria can swap plasmids like kids swapping Pokemon cards. When bacteria have what passes for sex in the prokaryotic world, they will pair up, and one will make a copy of a plasmid and pass it to the other one. Because sex at its most basic level is genetic engineering, mixing your DNA with somebody else's DNA. And so bacteria can swap plasmids. bacteria can pick up plasmids from the environment. And this is basically what was going on when Griffith did his experiments that showed the transforming principle. Remember, the bacteria that were harmless were picking up something from dead, harmful bacteria that was transforming them into more harmful bacteria. What they were doing was picking up plasmids from the dead bacteria. Eukaryotes have a defined nucleus bound by a nuclear membrane. In this fluorescent image, the DNA has been stained blue, so those blue blobs in the middle, that's DNA in the nucleus, and there's a clear boundary between the zone where the nucleus is and the zone where it isn't. The green dots and the red fibrous stuff are different components of the cell. We'll talk about those in a bit, but what I want you to see for right now is that sharp dividing line between the blue nucleus and the red and green rest of the cell. That's the defined nucleus bound by a double-layered nuclear membrane. This is a slice through a eukaryotic nucleus. It happens to be a plant cell, and most of what you're seeing is nucleus, and it's kind of light and grainy on the inside. And inside it, kind of down and a little bit to the right on this picture, there's a darker area called the nucleolus. The nucleolus happens to be the place where ribosomes themselves get made. The nucleolus is where that happens. Makes the nucleus look a little bit like a fried egg, with the nucleolus being the yolk. And then RNA, ribosomes, proteins, and things like that can get in and out of the nucleus through what are called the nuclear pores. What you're looking at here is it's kind of neat how they do this. It's called a freeze fracture image. What they're able to do is freeze a eukaryotic cell and then crack it. And when it cracks, the crack doesn't just split clean down the middle. It actually goes, it actually breaks in such a way that the crack runs over the surface of structures inside the cell. So you're looking at a three-dimensional picture of the surface of a nucleus. The big round thing is the nucleus. And if you look carefully, you can see that the surface of that round thing is speckled with little dots that look like, well, they look like my face when I was 13, but never mind that. They're speckled with little round things. Those are the nuclear pores. And the pores are kept open by sets of proteins called porins. The porins can open and close like little gateways to allow messenger RNAs to pass out, to allow ribosomes to pass out, to allow certain proteins to come in. The porins regulate what they call nuclear trafficking, just the passage of important substances in and out of the nucleus. And it's kind of hard to see here, but they can actually open and close a little bit like the blast door that Han Solo gets through in the chase scene on the Death Star in Star Wars Episode 4. That might help some people more than others, but there you have it. So those are the nuclear pores. If you look to the left and below of the nucleus, you can actually see some very fine parallel curves. That's part of the next thing I'm going to talk about in the cell, called the endomembrane system. And the endomembrane system has everything to do with all of the products that the cell has to make. If you want to imagine for a moment that a cell is a factory, you could think of the nucleus as the place where, the blueprints are stored. And the actual factory floor is a set of folded membranes called the endoplasmic reticulum. Reticulum means a little net. Endoplasmic means inside the cytoplasm. So the endoplasmic reticulum is the little net inside the cytoplasm. If that's still a mouthful, you can just call it the ER. And the ER you could think of as being a transport network within the cell, especially for cells that make things that have to be exported. Quick example that you've all got close to hand. You've got salivary glands in your mouth that make saliva. One of the things that's in your saliva, other than water and some mucus, is digestive enzymes. If you just put a dry cracker in your mouth and just let it sit there, it'll start breaking apart as it gets wet with saliva. And the reason is that you've got enzymes that start breaking down starches, which are crackers have a lot of. The enzymes in your saliva break those starches down into sugars. And one of the effects is that the cracker will slowly start tasting sweeter. And the other effect is that the cracker will fall apart. Well, you've got cells in your salivary glands that make this enzyme. It's called amylase. And those cells make it, but they have to export it so that it goes in your saliva, out of the cells into the saliva and ultimately into your mouth where it's needed. So the cells that make up your salivary glands have to make a lot of protein and then export it out of the cell. So they have a lot of endoplasmic reticulum. Proteins get made and other products get made via the ER. So you could think of that as being the main assembly lines in a cell. Proteins that get made on the ER often end up in a part of the system called a Golgi body, named after the guy who discovered it, who won a Nobel Prize back in 1905, Camillo Golgi. And you could think of this, if you like, as the packaging and shipping center, where proteins that are going to be exported out get sorted and packed up. Also part of the endomembrane system are vesicles and vacuoles. These are just sacs of membrane. Vesicles tend to be small, vacuoles tend to be big. They can be used for transporting certain things. They can be used for storage. They can be used for digestion in some cells. There's lots of things that these do, and there's no real structural difference except vesicles are small and vacuoles are large. And if you take a slice through a cell, this is what the ER looks like. It's a meshwork of folded membranes. Imagine you took a great big sheet, like a bed sheet, and you folded it several times onto itself. You'd get something that looks like the endoplasmic reticulum. This provides a lot of surface area for chemistry to happen. And in cells that make lots of protein for export, the ER is studded with ribosomes. All of those little black flecks that you can see between certain layers of membrane, all of those little black spots, those are all ribosomes. And in cells that make lots of protein, the ER is studded with ribosomes. It's just dotted with them, and it gives the ER a rough appearance. And so we call this rough ER. Ooh, ah, rough ER. Many cells have ER that isn't studded with ribosomes. This is also part of the ER. Looks a little bit more like a bunch of tubes, and this is also what the ER can look like. If you could see it in 3 dimensions, it would be this very complex network of folds and tubes and sacs and passages, all made of membrane. Smooth ER is not studded with ribosomes, and a lot of enzyme activity would go on in the smooth ER. Your liver, for example, is the main place where poisons get detoxified. If you take in a poisonous substance, it will hopefully get broken down by enzymes in your liver. If it doesn't, you might end up with a slight case of death. But liver cells would have lots of smooth ER because that's the space where this enzyme activity goes on. So cells that produce enzymes that break down poisons have lots of smooth ER. Cells that produce substances that aren't proteins like fats or carbohydrates, those would have lots of smooth ER as well. And it looks smooth, sort of, because it doesn't contain, it's not stuck to ribosomes, right? This, you can see a lot of the black specks that are ribosomes. This is not peppered with the same kind of black specks. This is a slice through a Golgi body. And again, you're seeing a cross section through here. You're not seeing a Golgi body in 3 dimensions. In 3D, imagine you took 10 beach balls, let the air out, and then stacked them one on top of the other. You'd have a model of a Golgi right there. You're just looking at a whole bunch of membrane sacs that have been sliced through. Up above, if you look carefully, you can see little circles above the Golgi itself. Those are vesicles, tiny little bubbles of this membrane. And vesicles are always fusing with the Golgi. Remember that membrane being a 2-dimensional film of liquid, little bubbles of it can always join together and can always break off, like the bubbles you could see in some nice bubble bath or dishwashing liquid foam. So the Golgi constantly has vesicles joining it and vesicles budding off of it. So things that are secreted from the cell, anything the cell has to export to the outside world, is sorted in the Golgi. It may have various little modifications added to it. Some proteins have carbohydrates stuck to them. Or they might get split in various places. There's a whole bunch of things that can happen in the Golgi called post-translational modification, which we don't need to get into. And anyway, then they bud off and form these little vesicles, and the vesicles can move to the outer membrane and fuse with the outer membrane, which releases the contents to the outside. Don't worry about the stuff in this diagram like constitutive secretion or regulated secretion or, synthesized soluble proteins for constitutive secretion. Look more at the picture that's going on. Don't worry about memorizing the words. You can see little vesicles caught in the act of budding off one face of the Golgi and moving to that outer membrane, and then fusing with it, releasing the contents to go to the outside. There's various ways that the process can be controlled that we don't have time to get into right now. If you want to change your major and take cell biology, you can get the details in agonizing detail. Right. Vesicles are these tiny little blobs. What you're looking at here is plant cells. The darker objects that you're seeing are organelles called chloroplasts, and I'll talk about those in a bit. That's where photosynthesis is actually happening. Most of the inside of a plant cell, though, is empty space. So in particular, in the center and over on the left a little bit, there is a cell that's got a big area that's just empty and doesn't seem to have anything in it. That's a plant cell's central vacuole that in life would be filled with water. Ooh, Actually, one of the things that central vacuole does is water pressure inside the vacuole gives the cell some rigidity. If you take lettuce, like next time you go to the grocery store, if you buy some lettuce, right, as long as it's moist, it'll be nice and crisp because the central vacuoles are full of water, and they're so full of water that they actually make the leaf a bit rigid, kind of in the way that a filled up water balloon actually has a certain amount of structural, stiffness to it. If you leave the lettuce out on the counter for, several hours, water will evaporate away from the lettuce. Water will end up leaving the cells. Water pressure in the central vacuoles drops, and the lettuce becomes floppy, becomes wilted. That's a result of those central vacuoles collapsing because they've lost too much of their water and no longer can support the cell. That's what wilting does to you. The fancy name for that kind of pressure is turgor pressure, but we don't need to get into that. But that's one of the things that central vacuoles do in plant cells that you could actually observe. Okay, surrounding the central vacuole, you've got chloroplasts, and I'll talk about those in a bit. You may also have structures called mitochondria. This big thing that looks like a sausage with stripes is a mitochondrion. If you look up and to the left above it, you can see a lot of rough endoplasmic reticulum, by the way. And apparently this is a meme these days as kind of an example of useless stuff that you learn in college. Mitochondria are the power plants of the cell. Apparently this is very funny when it makes the rounds of Facebook or things like that. Maybe I can go into a little bit more detail about why that actually matters. Most of the work of converting the food that the cell takes in into usable chemical energy that runs everything else, most of that chemical energy is generated inside mitochondria. I'll give you a little taste of how this works. There's a compound that your cells contain lots of called ATP, adenosine triphosphate. You might recognize, by the way, that it's a ribonucleotide. On the right, you can see you have that double ring, pentagon next to a hexagon containing carbons, but also nitrogens. That's a base, that's adenine. To its left, that pentagon with the oxygen stuck on, that's a sugar, that's ribose. And then on the left, you have 3 phosphate groups instead of just 1. That thing that looked like HOPOPOPO, hopo, 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 or whatever it is. That is 3 phosphate groups stuck one to the other. What ATP can do is give up one of those phosphate groups, which means it's now not adenosine triphosphate, it's adenosine diphosphate, and it will stick that phosphate group onto another molecule. So here on the right, you've got a molecule of adenosine triphosphate. What happens is that ATP comes up and it gives up one of its phosphate groups and sticks it onto the amino acid serine, turning it into phosphoserine. Who the hell cares? When ATP gives up a phosphate group, it conveys energy to the molecule that receives it. That serine that gets the phosphate stuck to it also has energy added to it, and the serine may be capable of participating in a chemical reaction now that it's got a kick. ATP you could think of as the fuel that runs metabolism, at least the reactions that are endothermic, the ones that require energy to run. There's a heck of a lot of chemistry involved here that we just don't have time for, and that I'm not the best person to teach anyway. We've got perfectly good biochemistry classes in the chemistry department if you really want to know. But when ATP gives up a phosphate to a protein called myosin, it causes myosin protein has changed shape. It can move a little bit and bond with a second type of protein called actin. Okay, howdy. You came in a little bit late. We've been talking about cell structure and talking about what structures like mitochondria do. The mitochondrion is the powerhouse of the cell. Phosphate to the protein. That enables the myosin to slide a short distance past another protein called actin. And that's how muscles contract. Myosin is able to repeatedly change shape and go ka-chunk, 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 sliding past another protein called actin. What makes it possible for myosin to change shape is that myosin picks up energy from ATP breaking down to form ADP plus a phosphate group. Okay, that is only the tip of the tip of the tip of the iceberg, but that gives you some idea as to what ATP does. Your mitochondria carry out most of the chemistry that takes your food and extracts energy from that food and uses it to turn ADP back into ATP. This is evidently someone here on the right whose body is very good at that. The amount of ATP that you break down into ADP every day would be equal to your own body's weight if it all existed at one time, but it doesn't. Every ATP molecule loses a phosphate to become ADP and then picks up a phosphate to become ATP somewhere between 500 to 750 times every day. You're constantly cycling through breaking down ATP and building back up again. Breaking down ATP is how you make chemical reactions happen that need energy, and then building ADP back up into ATP. Most of that work, not all, but almost all, goes on in your mitochondria. So this is why we say the mitochondria are the power plants of the cell. They're able to regenerate ATP from ADP plus phosphate by doing some absolutely gorgeous chemistry that we just don't have time for in this class. And then chloroplasts are the organelles where photosynthesis takes place. These are the things you can actually see if you look at a plant leaf cell as little green dots. This picture shows a water plant called Elodea. Normally, you'd get to see this with your own eyes in lab 2. I'm sorry we couldn't do that this go around. But these green dots are where sunlight is actually trapped. Solar energy is captured and used to build larger molecules that the cell needs. This is where photosynthesis happens. Plant cells have both mitochondria and chloroplasts, by the way. You do not have chloroplasts because you can't feed yourself by soaking up sunlight. Unfortunately, I guess. Right. What supports a eukaryotic cell is a network of proteins called the cytoskeleton. Cyto, by the way, means a cell. Cytoskeleton means skeleton of the cell. Duh. Right. And proteins, specialized proteins, make structures called microtubules and microfilaments. We don't really need to know how those work. Let's just say that they support the cell. They support structures inside a cell. They hold them in place. And in many cases, they will allow the cell to move. So the amoeba that we started with, or for that matter, your muscle cells or your crawling white blood cells, move because they've got a cytoskeleton of a particular type. These are pictures of eukaryotic cells where the nucleus has been stained fluorescent yellow and the cytoskeleton has been stained fluorescent red. You can't really see the cell membranes here, but you can see how the cytoskeleton extends to all parts of the cell. Those spindle fibers that we talked about way back when are actually a specialized part of the cytoskeleton. Finally, there are some eukaryotic cells that have hair-like structures sticking out, and they beat, and they'll either push the cell through fluid or they'll push fluid past the cell. This is a picture taken with a scanning electron microscope of some epithelial tissue from inside your lung, and it looks like a shag carpet. Every one of those cells has a number of little hair-like things, and in life those hair-like things constantly beat. What they do is push mucus. Mucus is produced in your lungs. It traps the dust particles that you breathe in, and these hair-like structures, as they lash back and forth, push the mucus past them up out of the lungs, up to where you can cough it up and then spit it out or swallow it. Those hair-like structures, if they're long, we call them flagella. If they're short, we call them cilia. Flagellum is Latin for a whip. Cilia is Latin for an eyebrow. So, like if you have a whip, you probably have just one and it's long. Your eyebrows are made up of lots of cells and they're short. But there's no real difference between the two except length. They've still got the same basic structure to them. Some eukaryotic cells, these hair-like structures beat back and forth to move the cell, like the flagella of sperm cells that enable them to swim, or the cilia of certain protists that enable them to swim. In other cases, the cilia move fluid past the cell rather than pushing the cell through the fluid. They work the same way. Okay, I could keep going, but this is actually a fairly good spot to pause. I know that's a lot of detail, but I'm going to give you something after a break that makes it make a bit more sense. So I'm going to pause this. I've got 9:50. I'll give you the usual 15 minutes, so you can come back at 10:05. Cats, or they're going to eat me. And okay, and I'm going to stop recording.